Thank you very much, Henry. I will try to be brief, and it's a bad time after lunch, so please excuse me. And then after all the contribution of the researchers and doctors and experts, it was very difficult for me to say something really useful for the profile. You have the background of the people here. But I think that really when we have a good outcome in science, then can open a new scenario that is maybe to create a new activity, a new business. So today I will present our experience as part of our journey from research to the market. Okay. So in our case, we had a starting point, if we go to the next slides, that started in 2014, and I organized a little bit for stages, at least the first phase of our company. If we go to the next slide, please. We see we were very young, actually. I met Luis when I met Artemisia annua plant. Luis was a young researcher, a colleague of my brother. They were both working in the CRAG, the research center in agrigenomics of Barcelona. And Luis had a very nice project with Artemisia annua plant. And with trichomes, you already knew the photo, the picture of the dragon took by Luis during his first researchers. So we met each other and we started talking. I was really passionate about Artemisia annua potential, and we found immediately a common purpose. If we go to the next slide, we could see the big potential of the plant, that it was very rich in more than 200 compounds from one side, and from the other side we saw the importance in the market of the artemisinin molecule. So we wanted to explore both paths. So from the beginning was clear we wanted to explore the plant, the whole plant. Okay. If we go for the next slide, we can see, but we identified, as we jumped in the market, 4 main problems connected with the plants. From one side, the low molecule concentration in plant, the lack of standardization in all the bibliography we were reading. It was very difficult to find a starting point with a standardized, let's say, ingredient or a compound that was comparable between different works. From the third side, the raw material we starting analyzing was very poor. The quality, the efficiency, the percentage was not clear. And finally, there were not really evidences of efficacy of the plant itself. The majority of bibliography was on the molecule artemisinin, and there was a few things on the plant. We are talking about 2014. Okay, let's go. So with all this information, we prepared the proposal. My previous job was fundraising for international cooperation, so we decided to write a project. The proposal was finally approved by the government of Spain, and this was the beginning of our adventure. So the first project was called the Tricopharming project. We had 3 main lines, which aim was to grow the concentration of artemisinin initially in plant. And they were 3 different paths: GMO, in vitro, and biostimulations. Guess what? The third one was the one that got the best results within this project. So the second part of the project was to jump from the lab to the field. So we identified Tenerife in Canary Islands as the best place to do it. We closed an agreement with the ICEA, the Center of Agriculture of Tenerife, And we made the Nagoya permission, so we made all the paperwork to introduce the cultivation of Artemisia annua in Canary Islands that was considered an invasor species. So it was very long, this part of bureaucratic papers, but finally we started 2 trials in 2 different latitudes of the islands, with the seeds that were the seeds that Luis was using in the lab. So we set everything. We were ready, but the seeds, first problem. So we are talking about a startup. We wanted to create a something with this plant. We didn't know yet what exactly. So everything prepared, the stations, the seeds, and the seeds did not germinate. So 1 month, 2 months, all the technical people, staff of the lab looking there, and there was no way. Finally, we had, maybe we discovered that we're like lab seeds, very used to controlled conditions, and then we had to change all the seeds. We bought from the York project, from the York University, and finally the project could go. So what were the results of the project of 2017? We are right. If we go to the next slides. Okay. With our biostimulant that we later will call tricostimulant, that Luis showed today also, we made field trials with a control group, not treated, and with biostimulants, and we got to a great average size and a greater trichome density. So this was, we made a lot of trials to confirm the results. And so this was, for us, a starting point of the real adventure. If we go to the next slides. So we entered in a program where we decided to generate like a brand of our optimized Artemisia annua. We optimize it through a process based on tricostimulant. And then we create the company. We create the company, and we were ready to start. But of course, we had to think about the market, who we are going to address this Artemisia annua so special. So if we go on the next slide. And then it was open like a Pandora pot, because we had like 200 references in bibliography. We tried to collect and to organize all the applications the plant could have. And in parallel, we started to analyze the market. In human health, we had the pharma and nutraceutics and cosmetics and traditional uses. Then in agriculture, we had a lot of possible uses also. We also had references as a natural conservative. Later, we made some tests, some trials about it. So we didn't really know. Of course, we wanted to start with human health. But since the beginning, we found a great problem in the European regulations. So we got a very bad surprise discovering that we could not sell in any format the plant in European Union. So we didn't have the capacity to go to the big pharma, of course, because we had very small volumes. So we started investigating the animal health sector that was really, really promising. But of course, we had all the indications for the animal health. So if we go to the next slides, we discovered that the real potential and the importance of the market in Europe could have centered in the leishmaniasis, that is a zoonotic disease. Very, it's a big concern in Europe. There are 2.5 million pets affected per year, dogs, sorry, then there are the cats. And we started a trial within a project that we had financed with the Institute of Tropical Diseases in Tenerife. and in vitro trials, showing finally the effect, the inhibitoring effect of Artemisia annua, Artenua rather, against the parasite from one side. From the other side, like an immunostimulation effect that we later demonstrated with another in vitro assay. And finally, this year, this is like a couple of weeks ago, we just published the paper about this study we realized in 2019. So if we go to the next slide, so we finally decided to focus on pet health market, mainly focused in leishmaniasis, but not only, with the product that is in format of capsules. Each capsule contains 200 leaves of Artenua, that is our optimized Artemisia annua. And we started to work in the Spanish market with vets. Surprisingly, we find a lot of vets, a few vets actually, already working with Artemisia annua. It was quite our job to make Artemisia annua known by the Spanish veterinarians, because it was almost unknown to the majority. So, also, we were interested in livestock, of course, because we were reading a lot about coccidiosis. But we realized we didn't have like the volume to go to livestock market, and the cost maybe for countries with bigger superficie and lower cost could be a real good alternative for animal. We can discuss about it. The thing that is sure that the trend in animal health is that the market is going up. It's a growing market. So the first thing we have to see when we launch a new activity, a new startup, is to see in which market we are going to stay. Cosmetics is totally different from pets. Pets are totally different from nutraceutics. So the first thing is to analyze very well which are the trends. The trends are clear. Consumers want natural products with scientific evidence from the other side. In all the markets we are talking about, the wool plant, this is important too. So if we go to the other slide, then I will show you very briefly today our reality in Tenerife. We have a small farm, a very small farm. We have 6 production sector and 1 R&D sector where we work with other medicinal plants. We have a drying facility with controlled conditions of temperature and humidity. Harvesting and all that is post-harvesting is manual. And the final outcome are dried leaves. We reached this year 1 ton in 1 hectare. The standardized content is 1.3. And this is really standardized because we measure each lot of production like 3 times with sampling. We work with the University of Laguna in Tenerife. And we also measure casein, eupatorin, and quercetin with the protocol that Luis did with the university in La Laguna and also with other research center. And we have a quality certification ISO 90001 and the ecologic certification. So all the production is totally ecologic. We don't use pesticide. We use only trichostimulant. And we have auxiliary plants for pest control. And that's it. Next slide. This is our team. We are very proud of our team. are like divided between Barcelona and Tenerife. I would say that when you start, we are a startup, the human capital is the first thing you have to take with you very strongly, because we had also some phases with the new people that were not entering in the culture of our company. It was very an hard time for us, but today we are very happy with our super team. We have a lot of people working in R&D. If we go to the next slides, this explains also how we could have, during the last 10 years, by the way, just yesterday we turned 7 years old. So for us, it's like a... Yeah, because if we go to the next slide, and then we can... As you know, the startups have like the famous Valley of Death, okay? Maybe you know that. So it's very, we were talking with some colleagues that had the same problem. You have to survive until you get to this point where you go in break-even. So to survive means to find strategy, mainly fundraising strategy, because even if you start selling, the cost, your operative cost, the personal cost, will be always higher than your sales. So when you go beyond 5 years, 6 years, maybe you are out of this death valley. And if you are in biotech sector, you need like 7 years to do it. Okay, it's like proven. If you go back, I would touch this, that it is very, very important for a small reality like a startup coming from science. So how to survive from a financial point of view. Sorry, but it's my role, so I have to speak about money. And Luis, no. So we had, if you look, we had like the first year until 2021, just public funding, mainly aid. So it's like non-returning funding, because this is the way, because at the beginning we do not have any commercial activity, we are not self-sustainable, and we need aid. If we have a component of R&D very strong, it's easier, of course, to have some aid. Then we started a phase in which we opened to private equity, so we started the first round of financing in 2020, and the second one in 22. And so this allowed us to get to today that we are almost in break-even point. So this is a very good news for us. So it's important when we start an activity to have a good mix between public and private funding, okay? Also, if it's possible to sail since the beginning, okay? The perfect mix is the three things. At the beginning, we had 80% aid and 20% sales, and now we are on the other way around. So it's the 80-20. So this is the ideal. Fundraising is a full-time activity in a small, you know, you are laughing because you know, and but from another side, it helps a lot the structure of a small company, because we are forced to have reporting, to be very precise and very organized in the paperwork, and this is very good for the long-term sustainability. So nothing. Yeah, we can go. Okay. And to resume, these are like some tips I would like to remark before closing. Before launching a business, maybe it can be a social business, maybe it can be a spin-off that some researchers of you would like to do, but in any case, we have to ask some questions. The funders have to ask some questions. And you have to close yourself in a room until you do not solve these questions. Otherwise, you are not able to continue. Is there any problem I can solve? So, which problem I could solve if I continue with my idea? Which are the main trends in the market I want to go, okay? So the one, consumers like natural products that are efficiently proved from a scientific point of view, yes. Who are my clients? So final users. In our case, final users are dog owners, for example. In other cases, can be women that use like creams for. So analyze very well which are my clients, final users, which are their needs, and important, are they willing to pay for the product? This is not because in the case of leishmaniasis, we observed that it is a very bad disease, a chronic disease, and the pet owners have to pay a lot of expensive treatment that have a lot of secondary effects. So when we could explain a product that is natural without side effects, it is cheaper than the actual treatments that they are available, of course, it's a good news for them. Of course, then you have to prove with that is working. Then USP, the unique selling proposition, in which I can differentiate from other products that there are on the market. That can be the format, the characteristics. In the case of Artemisia annua, and it is the content. On other characteristics, packaging, whatever, but you have to find your niche, your characteristics that differentiate you from the others. Regulation barriers, very important. In our case, we are now in pet sector for this. So my suggestion is one of the first things to look at. Can I use Artemisia annua for human consumption in my country? Yes, okay. So can I cultivate it? Okay, no. So it changes the story totally. Then discover key players, talk to people at the beginning, talk a lot to everybody that can help you to understand. Okay, I finish, and money to start. Okay, that's it. Thank you. 5 minutes. Okay, I finished. So I would like to know, to open some debate. If some of you, if we can go to the next one. No, this is the second phase, but it's not that really, because it's a scale-up phase. But we do not have the time to enter. If we go to the last one, I would like to know if you have some business idea. I see some young people, maybe researchers that want to be like entrepreneur. I was talking with a guy. I can see that he's a social entrepreneur, and I think it's a very important part of this story. Okay.
Thank you very much. Thank you very much, Chiara, and I'm sure we'll have some questions you want to ask. Yes? Pamela?
I'm so happy somebody got a business started on Artemisia. We have one in the U.S., but this is great. It's for a health thing that looks like it really is good. And years ago, I had done actually a market for veterinary products and my students, and we saw that there was a huge market available. So my question is IP. Did you patent the biostimulant, I'm sure, and EU, international patent?
In European patent. Yeah. And then now we are making the international extension, and we have to decide which countries are we going to. But EU patent was present in 2020.
Okay. And the last part, so obviously you've trademarked your products. You have trademarks. Yes.
Yes,
Good.
cream.
And what's your ROI, return on investment?
Sorry.
What return on investment? What do you anticipate your return on investment will be? When?
Return
Yeah,
on
yeah,
investment.
the ROI.
Okay, we had like we are still in break-even point. So the return of investment is expecting in the next years. Okay, yes, but we are on the good way.
Good.
Today, this year, we are going to sell like 50,000 units of product, and we started with 70 units per month the first year. So,
can you scale larger? Do you have to stay in the Canaries, or are you thinking elsewhere, or is all Canaries?
Yeah, we are already looking for scaling up the production. Already we scaled once. Yeah, because now we are in 9 countries. So we started just in Spain in 2020, and then we got Portugal, Greece, and Italy, and now we are starting with Latin American countries, making agreements with yes, Brazil coming soon.
Thank you very much. Last question by Romain, maybe.
Thank you very much, Chiara. It was very interesting, non-scientific, but extremely important for scientists. I have a question which is a bit scientific, so I hope you forgive me. I'm very interested in your Leishmania study. I wish I had read the papers, but I cannot read everything. Because the translation to human Leishmania, like the visceral Leishmania by Leishmania donovani, major, and mucocutaneous Leishmania is really a plague. It's also the same drugs, they're expensive, they're highly toxic. So my question is, when you said you used optimized Artemisia, like the spread Artemisia, of course you had the control of the non-spread Artemisia, was it less effective? And did you try artemisinin alone at the same concentration, like the content of artemisinin in your doped plant, was this effective?
Yeah. Maybe Luis can answer better than I. Sorry, but this is...
Yeah, I mean, don't worry about the fact that you didn't read the paper because it was published 1 week ago, so probably or 2 weeks ago. So you are updated, but you cannot be as much as data. So regarding the Leishmaniosis strain that we try is Leishmaniosis infantum, which is the one that is affecting mainly dogs. So we didn't try Leishmaniosis donovani or amazonensis. Yeah. But you know, the impact of Leishmaniosis infantum in human or even in cats is not as high as in dogs. That's why we focus on that. So we tried different levels of Artemisia annua with different concentration of artemisinin, okay? And it was very clear that as more concentration of artemisinin, better was the results regarding Leishmaniosis. But it was done in vitro, okay? We try also iron, because you know there is a big doubt if iron could increase the efficiency of Artemisia annua. It is somehow proof that in cancer, you know, induced ferroptopsis, so it's better to use it, but in Leishmaniosis it was not clear. We used extra iron and there was not an increasement, but it was just in vitro results. So no, we just tried different concentration of artemisinin, but within the plant. So what we did, we tried 3 different concentration of artemisinin. One it was 1.3 hours, other 0.7, and the other it was 0.1. And it was a really huge, correlation. So at least in vitro, it seems quite clear.
Thank you very much.